right. Previous studies have provided strong evidence that exposure to particulate matter, or PM, is associated with heart and lung disease and death. However, much less is known about how PM exposure might affect the brain. Recent findings suggest that air pollution exposure is associated with adverse central nervous system effects, such as cognitive declines in elderly humans and neurotoxic effects in animal studies. It is unclear, however, how these effects compare across locations with different sources of PM. Dr. Kleinman's study addresses this gap by examining PM exposures in several U.S. cities with different PM source profiles. Dr. Kleinman's project is valuable to ARB for a number of reasons. His study takes advantage of a large, multi-city project already funded by the Health Effects Institute, without which the current study would not have been possible. His findings are also relevant to California, as the sites include a location that's located in the LA basin. And finally, the results of the study provide insight into how PM exposure affects the central nervous system. Now, before I turn this over to Dr. Kleinman, I'd like to provide you with a little of his background. So Dr. Kleinman has been studying the health effects from air pollution exposures for over 30 years. He's currently professor and co-director of the Air Pollution Health Effects Laboratory in the Department of Medicine at the University of California, Irvine. He earned his PhD in Environmental Health Sciences from New York University and an MS in Chemistry from the Polytechnic Institute of Brooklyn. Dr. Kleinman has published more than 100 peer-reviewed journal articles on a diversity of topics, including the uptake and dosimetry of inhaled pollutants, and health impacts from acidic and non-acidic aerosol exposures and from mixtures of particles with other airborne pollutants. Dr. Kleinman is a consultant to the U.S. EPA Science Advisory Board, a member of the Board of Scientific Counselors for the Centers of Disease Control and Prevention, and chair of the California Air Quality Advisory Committee. He also serves as a chairman of the State of California's Scientific Review Panel on Toxic Air Contaminants. Now, it is my great pleasure to introduce today's speaker, Dr. Michael Kleinman.
Lori, thank you very much for the introduction. Obviously, you collaborated with my mother on getting that done. But well, I want to start out by acknowledging Arazu Campbell, who's been an important part of the studies that were done. And Arazu, while she was at UC Irvine, was the person who had the insight to say that we're doing all these exposure studies, we really ought to look at the brains as well as the heart, lungs, and other organs. And so I want to make sure that, you know, her contribution, and she's also done some of the analyses that I'll be presenting, but she was intrinsic in moving our lab into looking at those areas. Just to make sure everybody's aware, these are studies that were funded in part by the California Air Resources Board, and they wouldn't have been done without that important support. The mouse exposures, as Lori mentioned, were supported in part by the Health Effects Institute. And New York University and Mort Lippmann in particular, as the PI of that project, made it available to us so that we could get brains from other sites in the project, as well as the ones that we were obtaining in Irvine. And of course, the California Air Resources Board takes no responsibility for the things I say. So, having said that, as Lori mentioned, the environmental effects on the brain may be important to study, we know they're important, but it's really important that we begin to study these things because the incidence of brain disease seems to be increasing. If you look at the statistics, they're constantly going up. The role of environmental interactions in this has become much more apparent as people start to look at epidemiological studies where you examine twins from families that are prone to developing Parkinson's disease, where one twin is raised in a community with low levels of pollution, others are raised in higher levels. And there seems to be, if nothing else, a delaying effect if you live in an area that has less environmental impacts. The mechanisms are still elusive, and what we are going to talk about today will be more in the phenomenological range, but we do have some things that give us some mechanistic insights. I think this field really got its impetus when researchers in Mexico found that the brains of, I believe the first studies were done with canines that were roaming Mexico City. And when they looked at the brains of those dogs versus the brains of dogs in cleaner communities, they found more brain lesions and damage in the animals in Mexico City. Subsequently, they performed studies with human volunteers who, not volunteers, human samples from accidental death victims, and saw much the same thing as we're seeing in the dogs. Now, the dog studies were always criticized because you didn't know where the dogs were, what they ate, very little about them. The people they had a little bit more control over. And then as scientists began to see this as a real phenomena, better epidemiological work was done, and all of which seems to support those original suppositions that high levels of air pollution are related to damage to neurological and central nervous system tissue. There is some evidence, and I'll show you some of that, which actually came out of the NYU studies and some of the earlier work that led to this work, that shows that there are definitely effects on the critical parts of the brain that are responsible for making dopamine. And that's where the link to Parkinson's may be. Annette Peters in Germany collaborated with the NYU team and did histological analyses of many of the samples that were obtained from animals, and she also did work with humans. And again, she found that there was a link between PM exposure, and it probably implicated oxidative stress as the overall mechanism of damage. The other factor that she pointed out was the data are beginning to support the idea that particles that are inhaled can translocate out of the respiratory tract and reach distal organs. We certainly know that there are effects on the heart, and in Annette Peters' work, there were clearly effects on the brain tissue. Now, when you start to think about how to do environmental studies, on the brain, it's extremely difficult. If you're working with environmental concentrations, even though we consider these levels and we set standards, and in many places the standards are being exceeded, but the levels are very low compared to occupational levels, and here in the U.S., certainly lower than what we see in some of the capitals of countries like China and India. So how would you get enough of a dose? Because this is all dose related. How do you get enough of a dose into an animal in a reasonable amount of time so that you could actually see some of these things happening? So one approach is you expose for really long periods of time. Another approach would be to use huge numbers of experimental subjects. And both of those approaches are not terribly practical. Most funding sources are, 3 to 4 to 5 years. And you just don't get those kinds of results usually in a short term. So we have to engineer the experiment. So what I'm going to show you is data based on real-world atmospheres, but in order for us to see the effects in a reasonable amount of time, we've used a particle concentrator to increase the concentration of particles. Now, I'll show you that in a minute, but I just wanted to point out that even though we're using this mechanical device, or this engineering device, as our way of speeding up the process, the concentrations we're using are not unbelievably huge. So we are typically exposing animals at about 100 micrograms per cubic meter, roughly 10 times the local ambient levels, well below what you might see in Beijing on a good day. So I was joking with Lori, if I had a colleague, or with Linda too, if I had a colleague in China, I wouldn't need the concentrator. I could just bring the rats over and do it. But one of the things that have eluded us, because it's difficult to get this kind of data, very few researchers use exactly the same protocol in their research. And that was the wonderful thing about this HEI project. We had several cities, a team of investigators. They decided on an exposure protocol. All of the equipment was matched so that our exposures were done in an equivalent way with an equivalent time period. And we tried as best we could to match up on the biological endpoints. And that allowed us to do real comparisons between other cities as well as cities in California. So our objective was to determine the biological effects in the brain from mice that are exposed to fine particulate matter. Now, fine particulate matter, we're talking about PM with a upper size cutoff of 2.5 microns, or micrometers. And that's the definition of PM 2.5. It's the particles smaller than 2.5 microns. When people look at this and they start comparing it, they say, well, okay, so these are particles that are 2.5 microns. No, they're particles smaller than, and in fact, when you measure it, you're really talking about particles that are about 700 nanometers on the average. So even though we have this upper limit, only a very small percentage of those particles are really up at that large size. Most of the particles are small. And they come from motor vehicles, they come from power generation, industrial processes, even things like road dust being resuspended. A small fraction of the road dust is still in that fine particle category. So we wanted to address the question, are PM-induced biochemical changes on the central nervous system going to be influenced by particle concentration or by the chemistry of the particles? And as a chemist, I was voting for chemistry of the particles. We're looking at in vivo biological responses, and we tried to pick responses that other people have also identified as biomarkers of potential central nervous system exposure or injury. And as I said, we had 5 different cities. Each of those cities has a very different source profile. So this gave us the opportunity to leverage resources very effectively. So let me tell you a little bit about the exposure design, and then we'll talk about the cities themselves. But we had mice that were exposed in New York City and really Midtown Manhattan, Sterling Forest, New York, which is, as the name implies, in a forest. It's a very rural area and very isolated from city, even though it's only about 50 miles northwest. So you go 50 miles out of New York City and you're in this, what looks like a pristine wilderness. Lots of trees and dirt roads and things like that. However, because of where they sit, they are influenced by regional pollutant sources as far away as the Midwest. So in each of these cities, Seattle was another one. We got samples from East Lansing, Michigan, which is near Detroit, and Irvine, California, which is an area that most people don't think of as a polluted area, and it isn't, although our levels are creeping up over time as the population grows. But as I'll show you, we are influenced by a number of nearby sources. The mice were exposed for 6 hours a day, 4 days a week, for a total of about 26 weeks. Now, what that means is the animals are housed under purified air conditions most of the time. For 6 hours a day, they're brought out and placed into exposure chambers and exposed to ambient particles. At the end of the experiment, or the exposure phase of the experiment, brains were harvested, and then various biochemical analyses were performed. So we looked for inflammatory cytokines, interleukin-6, interleukin-10, tumor necrosis factor alpha. These are all 3 biomarkers that are known to be associated with inflammation in any organ. All organs tend to have the same inflammatory mix, driven by different factors, but these are really good markers for when you have an inflammation going on. We looked at reduced and oxidized glutathione because these are markers of antioxidant defenses. And usually, but not always, when we see inflammatory changes, they're accompanied or even preceded by oxidative changes and oxidative stress. And then we looked at biomarkers of oxidative stress. So if your antioxidant defenses are overwhelmed and you're generating a lot of oxidants and free radicals, these will react with proteins and lipids, and they will form other products that are relatively long-lived and can be measured. And so things like protein carbonyls, hydroxynonenal, and malondialdehyde are all oxidation products from proteins and lipids. So when you see those, you'll see those in areas where your normal defenses have been overwhelmed, or at least not able to keep up with the oxidant in the short term. Particle samples were collected while we were conducting the studies, and so day to day we collected air filters. Those filters were analyzed and we looked at the total mass, we looked at the trace elements, and we looked at the elemental and organic carbon contents. We then had all of this data, and as I'll show you, one of the neat things about having this trace metal data, or these trace element and compositional data, is it can be used to give you hints as to what were the sources of the PM that the animals were being exposed to. So what I'm not going to show you is a formal source apportionment study. They actually did some of that at NYU, taking all of the data from all the sites, and they did some. I'm going to show you more of a conceptual idea using key tracers. And that may be, yeah, I think that's a good way of looking at this, just to see which sources were mostly influential about producing the particles that the animals breathed. Now, all of the animals in this study were ApoE knockout mice. That means that they have a genetic imbalance. They don't metabolize lipids properly. And so these mice all have very high levels of circulating cholesterol, especially low-density lipoproteins. As you might imagine, these animals are very prone to developing atherosclerosis, and they were the animals selected because the main HEI study was primarily a cardiovascular study. So we slipstreamed on the cardiovascular study to do the central nervous system tissue. So mice were either exposed to filtered air or to concentrated ambient particles, or CAPs. And we did studies for 2 months, 4 months, and 6 months. So you can imagine at the beginning of the study, when we had all 3 exposure groups running at the same time, we had a lot of mice. All of the all the protocols were approved at the different institutions by their various animal care and use committees, and that's really important because everybody had to adhere to the protocol. It was really, we all had the same basic protocol, so it was a good way to ensure that we're going to have comparable data. We had a total of 16 mice in each exposure group, and 6 of the mice in the of each exposure group were implanted with telemetry devices. Now those stayed in the experiment for the full 6 months. And the implanted mice were monitored for electrocardiographic changes. And we could look at them both during exposures, after exposures, and on days when they're not being exposed. And so a great deal of data has been generated, and we're still analyzing a lot of that. While they're in their housing, they get purified air. They have food and water. One of the sort of experimental problems with doing these studies is the 6 hours a day that they're in the exposure chambers, they don't get to eat or drink. So these animals are probably at a slightly higher stress level than they would be if we compared them to cage controls. So all of our controls go through the same level of stress. They're all treated the same. So overall, we had about 22 hours a day of cardiac monitoring data, but we only got neurological data at 2, 4, and 6 months. And unfortunately, there was no behavioral component to this, so we don't know whether there were any behavioral changes. So the sites. New York City, as everybody, I'm sure, is aware, is very densely populated. It's an urban environment. The major impacts are traffic, and it's one of the cities where, right in the middle of the city, you have these tremendous oil-fired power plants. And these are running 24/7. When I was living in New York, in addition to that, you had incinerators and all sorts of other things going on. But those have been eliminated. So the biggest sources are the traffic, the oil-fired plants, and the dust kicked up by all the vehicles on the road. So it's not just tailpipe emissions, it's tailpipe emissions plus all of the other stuff that cars generate. Sterling Forest, as I mentioned, is a rural location. It's got a lot of regional source contributions. And what I hadn't realized when I lived there, I actually lived there for a while when I was doing my doctorate work, that there was such a big influence from the Midwest and particles from coal burning in the Midwest. However, they've also found since then that on rare occasions there are some nickel smelters in Canada that put out large amounts of nickel aerosols, and on certain days those would blow into Sterling Forest. And because there wasn't very much other in terms of specific local sources, Lippmann and his colleagues actually were able to show a significant effect on cardiovascular effects that they associated with the nickel from these smelters. And that sort of plays a role because nickel and vanadium are key chemical components of oil used in power plants and also on ships. And so when we start looking at the trace metal data, one of the things we found was even Irvine, which is, we were thinking, relatively clean and unpolluted, we could see influences of oil burning, and mainly the main sources would be either diesel traffic, which usually burns cleaner fuels than fuel oil, or the ports, which are somewhat north and a little bit west of us. But on certain occasions, I'm sure we get winds blowing from those directions. And it was enough there that we saw an influence. So the ambient particle concentrator, this is a device that was developed by Siotis and his colleagues at USC, and were an outgrowth, I think, of his work when he was at Harvard. So there's an East Coast, West Coast conglomeration here. But they developed a portable particle concentrator, and they nicknamed it the VACES, the Versatile Aerosol Concentration Enrichment System. And it's been very well characterized and very well published, and it's been extremely useful. So this is what a schematic of our exposure system would look like, and the VACES plays a very prominent role. The VACES in principle is a fairly simple device. You suck in ambient air, and we have a stack on our roof so that we're not getting material from our parking lot or off the ground. It's drawn down through the stack and enters a tank which is filled with saturated water vapor in air. So the particles come through, mix with this massive wet air, and then is drawn up through these tubes that are wrapped with cooling coils. And we cool the air in the tube down to freezing levels, Well, that causes all the water vapor to condense onto the particles, because that's where the surface is, and those particles grow. And when they grow, they have a lot of inertia. And so we funnel those particles into a beam, and the beam comes up through a slit and into a pipe. And this pipe is drawing at 5 liters a minute, but most of the air is drawn off at 300 liters a minute. And we have actually 3 pipes, so you have a total of 300 liters a minute coming in, 15 liters a minute coming out. And all the particles are pushed into this 15 liters a minute. So the concentration is increased approximately 20-fold. And that's how we get the particle concentrations. Of course, they're wet. We have saturated water vapor. So we run it through a silica gel dryer. The particles dry out, and they actually return to very close to what the original size distribution was. And so we've done chemical analyses on the particles, we've done size distribution analyses, and we're very comfortable with the idea that what we're exposing the animals to are really concentrated particles, very similar in characteristics to what they would have been in the ambient air. And then they come out and we go through analyzers, but most of the particles go into our exposure chambers. And in the exposure chambers, they're distributed, and the animals are breathing a relatively uniform amount of particulate matter. And in this particular case, we were running, it was nominally 100 micrograms per cubic meter. However, different cities, even though we followed the same protocol, there were some differences. So first of all, time differences. The studies in New York and Sterling Forest were done quite a while ago, in 2007. Seattle was done in 2009, and then Michigan and Irvine were done 2010, 2011. The Seattle study was done during the winter to take advantage of the fact that in the Seattle area there is, during the winter, a lot of wood burning for space heating. So a lot of people have wood burning fireplaces, wood burning stoves. And it's one of the prominent sources of air pollution during the winter in the Seattle area. And we thought that would make that very different. So they have some traffic there, but it's not as much as Southern California or New York, but they have a lot of wood burning. So if we look here, the ambient concentrations in micrograms per cubic meter, and this is a mean, and then it's followed by a standard deviation, so you can get an idea of how variable it is. But New York and Sterling Forest actually had the highest ambient levels. That probably had, spoke to the fact that these were done in 2007. Pollution levels in general were higher then than they are now. So that may have been part of it. New York City always has had high levels of PM. Irvine, our clean city, was about the next highest after those two, but not very different from the total in Seattle or East Lansing. But when we concentrated the particles, we actually had higher concentrations in the Irvine particulate than in the other cities. When I set this up, I designed it to try to mimic the concentrations that we would see in what they saw in New York. That was sort of my target. And we were able to get up, and as you can see, we were pretty close, and certainly within the standard deviations. The other cities, their concentrators, for one reason or another, didn't work as well. So their concentration factors were a little bit lower. But the other thing I wanted to point out, when you start to look at the composition, the chemical components, so BC is black carbon, EC is elemental carbon. So black carbon is the way to characterize sort of overall soot, and it's elemental carbon plus organics for the most part. And then we had just organic carbon. And you can see that Irvine and New York City had very high levels of organic carbon, pretty high levels of elemental carbon, or black carbon, and elemental. One of the things that were a problem in drawing some conclusions is, unfortunately, the elemental and organic carbon data from Washington, they weren't able to collect it. Something happened with the machinery or whatever, and they did not get enough data. The other thing I want to point out is when you look at all of the things that were measured, so we had several different trace elements measured. And what we did with that data, or actually the NYU team did with this data, was they ran statistical analyses. Now, what you're looking at on this table are averages over the 6 months, but those are averages of 4 days a week for 26 weeks. So a lot of data goes into this. And what was done was they did an analysis of correlation. So they did a correlation matrix of each of the elements with each other for each of the individual cities, and then used that to see which set of data correlated best with the PM concentration. So it gave you an idea that when you found something like nickel representing a substantial part of the variation, not substantial part of the mass, but when you plot the changes in concentration over time for PM in New York, and you plot the changes in concentration of nickel in New York, they lined up. They were highly correlated. So that would indicate that the source of the nickel was also possibly the source of a substantial amount of the particles. So that's sort of the premise of this. So using these strengths of these correlations, they were able to make some suggestions about what were the important contributors. So elemental carbon is a good tracer for traffic. And so it turns out that when you look at elemental carbon, the strong correlations were found in New York City and Irvine. When you look at nickel and vanadium, the highest association was in New York City, but we also found some in Seattle and a fair amount in Irvine. Crustal material, the road dust and things like that, are very similar to the Earth's crust. That's why they call it crustal. And aluminum and silica are good tracers for that. And they were very important in New York City, Sterling Forest, and Seattle. Now, those elements are usually associated with the bigger particles. But as I said, even those very large particles, a small percentage of that shows up in the fine particles. Wood smoke contains a lot of potassium. And the potassium signature was very strong in Sterling Forest and Seattle. We also saw some in Irvine. But looking back at the data for the time that we did the exposures, it turned out that for part of that time, there was wildfires going on. And so we may have seen an influence of some of the wildfire smoke as well. And industrial emissions and coal emissions have a fair amount of arsenic and selenium. And the selenium turned out to be a really good tracer, at least in this mix. And it turned out that New York and Sterling Forest and Michigan State, or East Lansing, Michigan, all are impacted with coal burning and all showed a strong signature to selenium. So I put this on a graph just to sort of give you an idea of what it would look like. And you can see the elemental carbon signature, very prominent for New York and for Irvine. Again, Southern Californian cars are incredibly wetted, so we expected that. The nickel and vanadium are signatures for oil, and I had not initially expected to see as strong a signature for Irvine, but it was there, and a little bit, and some for Seattle. We don't see as much road dust as is seen in some of these other cities. Seattle clearly had more potassium, and I think by experimental design, we were weighting the thing and expected to see a wood smoke influence. So I'm gratified that we see it. The coal and industrial, we're seeing in New York, Sterling Forest, which, as I mentioned, gets influenced by the Midwest. And actually, it used to be a mining area, so there could be residuals in their soil. And Michigan. So we were hopeful that, by doing this, we were able to at least make some statement about that the sources were really different in the different cities. Now, the New York City and Sterling Forest brains were actually analyzed before we got this project started. So we couldn't do very much with it other than look at what they found. And to some extent, we used their findings as sort of a guidepost for some of the things that we wanted to do. They did some histology, which was really intriguing. And they stained brains for cells that were high in tyrosine hydroxylase. Tyrosine hydroxylase is an enzyme that converts tyrosine through a chain of things, eventually ending up as dopamine. So cells that have tyrosine hydroxylase are probably dopamine-producing cells. And in this case, neurons. And we also looked for glial fibrillary acidic protein, GFAP, and this is a marker for inflammatory cells in the brain, the astrocytes. So it turned out that when the group at NYU and Annette Peters, I think, was part of this, a few other people actually did a lot of these analyses, they found about a 29% reduction in the neurons that stained for tyrosine hydroxylase in the CAPS-exposed animals compared to the filtered air. And increase in the inflammatory protein, GFAP. So this is a graph that was published and of animals exposed in a similar way, and they integrated the little black dots for tyrosine hydroxylase and found that the air exposed had much higher concentrations compared to the particle exposed, whereas the GFAP was higher in the particle exposed than the air exposed. So definitely these exposures can lead to damage to the part of the brain that makes dopamine, and so it does substantiate or at least strengthen the potential link to Parkinson's disease. So, and there's the suggestion, since this was looked at in the same area of the tissue, that the inflammatory response was part of what was going on with the reduction in dopamine. We took a little different approach. We were looking more at a regional analysis. And this is one of ours who's, shall we call it a brainstorm. She really thought this through and decided it would be really important to know something about what was going on in different parts of the brain. And so when we whoops, stop. I don't know how I did that. Sorry. So when we harvested the brain, we cut it into 3 sections. The rostral section, which is in the front of the brain, the anterior part, the midbrain, and the back of the brain, the caudal section. And then we looked at these animals at 2 months, 4 months, and 6 months. So we looked at inflammatory cytokines, interleukin-6, interleukin-10, TNF-alpha. And over time, we didn't really see a consistent trend. In fact, as I'll show you, we seem to see more of an effect at the beginning and end of the experiment, and in the middle of the experiment, it wasn't significant. But there were real significant differences as a function of region. So here are the 2-month, 4-month, and 6-month data. I think I build up too much static electricity and I'm affecting this thing. I shouldn't be trusted with equipment, but so interleukin-6, interleukin-10, and TNF-alpha. So the first thing I want to point out is when you look across the brain, you seem to have, in the exposed animals, a lot more of inflammatory protein in the rostral section than in the others, and it seems to follow a decreasing trend. And at 6 months, you see that decreasing trend. The unexposed animals almost show an increasing trend. So there's definitely an interaction here between front of the brain, back of the brain, and the development of inflammation after you've exposed them to particles. The IL-10 didn't show that. It showed sort of an increase as you go back in the brain, and really the differences between the air and the particle exposed were not significant. TNF-alpha followed the same pattern as the IL-6, and they usually are pretty well correlated. And at 6 months, there was a very significant increase in the rostral. Now, one of the things about these inflammatory cytokines is you want to ask, does that make sense, and where does it come from, and why should there be regional differences? So one of the things that is important in terms of where do these things come from and how they're made is signal transduction. So NF kappa B, nuclear factor kappa B, and AP1 are transcription factors that respond to oxidative stress and inflammation. And the way they respond is by getting phosphorylated, and in their phosphorylated state, they can migrate from the cytoplasm of the cell into the nucleus. When they're in the nucleus, they go to promoter regions of certain genes, and these happen to be the genes that make cytokines like TNF-alpha and interleukin 1 and 6. So activation of NF-kappa B should be related to increased levels of cytokines. And when we looked at whether air pollution can affect these things, this is kind of a dose-response study. So we used the concentrator and we modified it so that we had a low amount of concentration, and it increased ambient concentration fourfold, and we had a higher amount, and we got it to 15-fold. So you can see that AP1 shows a very clear dose response. NF-kappa B, these aren't significantly different, but the high level did provoke a significant increase. And so you'd also expect that you'd see more of these inflammatory cytokines when you have more activation of the NF-kappa B. So we looked at the NF-kappa B levels in the brains, in the rostral, midbrain, caudal sections, and we found more NF-kappa B activity in the rostral section in mice that are exposed to the particles, which kind of correlates with what we were seeing before. So it was consistent with the increased levels of the IL-6 and TNF-alpha that we saw in those rostral sections. But then we looked at protein carbonyl. Now, protein carbonyl is a marker for oxidative stress. And we found the opposite effect. We found an inverse relationship. So here's the NF-kappa B levels. And in this case, we're not seeing a big effect of particle exposure. So the air and the NF-kappa B levels were pretty much the same for, or not the air and the CAPS levels were the same for NF-kappa B, pretty much at all the time points. But there was more NF-kappa B in the caudal section than in the rostral or the midbrain. When we looked at protein carbonyl, this was only significant initially at the 2-month level. But we see protein carbonyls tended to decrease from the rostral to the midbrain to the caudal. So there may be other roles for NF-kappa B, among which might be in some way aiding in antioxidant defenses. So we wanted to look at site-specific differences in brain responses. And we looked at the Seattle, the MSU, and the Irvine sites specifically because these were all done fairly close in time, and we had more control over the tissue from those experiments. So we actually had enough tissue that we could do these analyses. So as I said earlier, the reduced oxidized glutathione is an indicator of whether exposures are affecting the antioxidant capacity. So if your glutathione level goes down and your oxidized glutathione level goes up, that means that there's not enough glutathione, or there's less glutathione to react with free radicals, and therefore more prone to oxidative stress. So we found slight differences in Seattle at 6 months and at UCI, but it wasn't statistically significant. But when we looked at the Detroit samples, the Lansing samples, the air and the exposed animals were exactly the same. But they also had much higher levels than the other two sites. So there were definitely differences in the mouse populations. The hydroxynonenal levels were increased at Seattle, but we didn't see significant differences in Lansing or Irvine. Malinaldehyde, which is an indicator of lipid peroxidation, we found those were generally lower after CAPS exposure. And at this point, all I can say is that was unusual, and we can't really explain it yet. But we're doing some more experiments, and hopefully we'll be able to find something about that. And then the protein carbonyl levels increased in the whole brain samples in Seattle, but not in Irvine. So putting it on a graph, you can kind of see here that the CAPS exposure reduced the antioxidant potential in Seattle and in Irvine. Now, you can see here that the error bars are bigger in Irvine. The reason for that is we had cut the brain into 3 sections, and so we analyzed the 3 sections, and then we had to add the data together, and when you do that, you have much higher variation. So the variances were high, so the trends are the same, but we can't really call statistical significance on it because the error bars are bigger. Hydroxynonenal was increased in Seattle, but not in either Irvine or East Lansing. And protein carbonyls, which is another good indicator of protein oxidation, again went up in Seattle, but a little bit, but not significantly, in Irvine, and nothing going on in Detroit. So, conclusions. We saw inflammatory changes in the brains. On a regional basis, we see that the sections of the brain that were lower in the signal transducer, the NF-kappa B, seem to be more susceptible to the inflammatory changes. And that's going to call for a lot more research on what is the role of NF-kappa B in inflammation. Looking at the time from 2 months, 4 months to 6 months, this is a substantial fraction of the animal's age. And so part of what we may be seeing superimposed on our exposures is an aging effect. So NF-kappa B tended to decrease as the animals got older, which might mean that as we get older, our susceptibility to inflammatory changes in the brain may be increased. And so this should be looked at probably in epidemiological studies where some of this can be examined. The part of the brain with the low levels of NF-kappa B showed increases in oxidative stress, which might be, which seem to be consistent with the inflammation oxidative changes. I think it's interesting, though, that what we're seeing is that the city with a lot of industrial input, East Lansing, but without the input from oil sources or traffic, or with less impact from those, did not have as much of an effect on the brain. However, when we looked at cardiovascular effects in the same group of animals, they had substantial cardiovascular effects as well. So different mechanisms are also going on. So the concentrations of nickel and vanadium in the exposure atmosphere in New York City and in Irvine, even though there might be an association with the oil burning sources, when you look at the absolute concentrations of nickel and the absolute concentrations of vanadium, they're much too small to have been causal in and of themselves. So they're pretty, it's very likely that they would be surrogates for something in the soot or the emissions from those sources. And I think that the data that we see says a lot about how the products of oil combustion from mobile sources, power generation, even space heating, these may be important factors in inflammatory and oxidative changes in the brains of these mice. And again, I want to point out that these are ApoE knockout mice. They are not normal. They have very high cholesterols, which mean when we looked at their background level of sort of systemic inflammation, those were higher than they would have been in the wild-type mouse. So does that, would we have seen these effects in the wild-type mouse? We're doing some studies now. Maybe we'll be able to find that out. But I think that it's an important question to ask. However, the other thing about the ApoE knockout is just, if you picture the arteries in the heart, which are fairly large, and they got plaque developing in them, the arteries in the brain are also potential targets. And if you block the arteries in the brain, you block oxygen delivery to the brain, and a lot of the same inflammatory changes that we're talking about here. I can't speak to the effects on the dopamine-producing cells, but the inflammation, oxidative stress stuff is something you would see in an anoxia sort of situation. So if you expose animals to very low oxygen tensions, you'll see inflammatory changes in the brain also. So it could be that we're seeing a number of different factors in these animals. So I just want to acknowledge, first of all, that we got a lot of help from the group at Air UCI, Barbara Finlayson-Pitts and her team helped us with a lot of the analytical chemistry that we've done on the particles. We did make measurements of the particle sizes, so we had some idea that what we're delivering to the animals was a reasonable amount, and that was different from what the other sites were able to do for the HEI study. Certainly the California Air Resources Board for their very strong support and their help. And then all of the collaborators that worked on the project, Mort Lipman, Lung-Chi Chen, Terry Gordon, and Arazu Campbell, Jack Harcum and Jim Wagner in Michigan State, and the folks in my lab that have really worked very hard to collect all this data and analyze it. So with that, I'd like to stop and answer questions if there are any.
wonderful talk. We're now going to open it up to questions, and we've already gotten one, or several, off of the web. So if you'd like, I can read the questions and hand it over to you.
This is a multi-part
This
question
is a multi-part question.
from Roger McClellan, who is probably one of the top experts on respiratory and air pollution health. So none of the changes in the brain are quite minimal, or the changes in the brain are quite minimal. How do you exclude the possibility that the effects, including oxidative effects, are a general reaction to the exposure to the pollutant mix and the absence of material already reaching the brain? Has non-reactive particulate aerosols like pure carbon black or titanium dioxide of equivalent of multiple sizes been studied for brain effects? I agree. The changes that we're looking at are very small. And I think that the exposure to the pollutant mix, one of the things about the concentrator is that it simplifies the mix somewhat in that most of the things that are present in the vapor phase do not get concentrated. So the particles are concentrated, and all the things that are attached to the particles are concentrated. But a lot of the organic vapors that are not stuck to the particle don't make it into the system, at least, or if they do, they're not concentrated. Other than that, we try to keep the animals, when they're not in the exposure system itself, breathing purified air. So I think we don't have a pollutant mix as if it were a pure ambient exposure. I think that would be the answer to that. And we've not looked at carbon black or titanium dioxide. At least, we've not done it. I believe some of the folks in Rochester have been doing studies with titanium dioxide and carbon aerosols. And I don't remember at this point what they've published. Allison Elder has been doing a lot of work on this, and I just don't remember whether they've seen similar effects. Okay, the second question was, were the investigators and technicians making observations on brain specimens blinded as to whether the animals were exposed or controlled animals? That I can answer. Yes. We did not, all the data were blinded at the point of analysis, and after we got the analytical data, then we opened it up to which group did they fall into. And then, in fact, we then put the data back to individual animals to see if there were any peculiarities within any one animal. Did that animal, seem to be ill or something like that, in which case we might have wanted to eliminate it. Now, I'm speaking for, the brains only. That a lot of the other animal handling and the way the tissue was treated was done at 5 different places. I'm pretty sure everybody had the same blind strategy. Were the animals coded and reread by other investigators? Again, for histology, we didn't do histology on our samples, and Lungchi Chen and the NYU team would have to speak to that issue. We did mostly biochemical analyses or electrophoresis studies, and those were, as I said, done blind and then reorganized. But once the data were reorganized, the numbers were the numbers. We didn't go back and we didn't have multiple samples to do. Whether these are, he's interested in whether the studies are being replicated with outbred or inbred strains of mice or of rats. We're currently doing a study on for a different project, but it's going to run for a year. And we're just using normal BALB-C mice, and we will be harvesting the brains from those animals. The goal of that study is to look at potential changes in the brain related to cancer formation in conjunction with Cedars-Sinai Hospital. But we'll be able to look at a lot of the inflammatory things as well. So I think the answer to that question is yes, they will be replicated, and in fact for a longer period of time, but we're not going to get the multiple time points. And I agree with his statement that the genetically manipulated strains are not particularly well suited. Again, we chose that strain because the brain wasn't the target of the original project. It was cardiovascular. Earlier in my talk, I noted an increase in degenerative neurological diseases. Do the alleged increases hold up on an age-adjusted basis? It is obvious that many of these diseases occur late in life or later in life, and with a population that lives to increasingly older ages, there will be an increase in the occurrence of these diseases. I completely agree that that's the case. I think the point I was making was that there's, in the epidemiological studies where they have been looking at samples or people from different communities, there seems to be a relationship to areas with more environmental contamination versus areas with less. Whether they looked at a whole age spectrum, I don't know, and I think it's certainly a very good question to ask. Yeah.
Johan Herner with CARB. Thank you for a very nice presentation. So your model was to expose rats and then dissect their brains, I guess, to look at them. I'm not an expert in this field, but I was wondering what your opinion is about using chemical assays for determination of toxicological effect of PM, the DTT, macrophage ROS, AIMS, all those things.
You mean in vitro tests?
Correct.
I think those are certainly useful, and we've been able to do some of those things. For example, we harvest macrophages and then test functional activity of the macrophage from exposed and unexposed animals. So that's, I feel, a nice way to use those kinds of data. For screening purposes, where you want to compare specific chemicals or particles from specific areas, it's a good first step. And you can certainly look at a lot of mechanistic things that way.
Hi, I'm Chris from Research Division. I've heard it argued that if ambient particles are affecting the brain, it's got to be the ultrafines that would be able to penetrate into the brain, 100 nanometers in diameter or smaller. And I'm just wondering to what extent you would agree with that statement based on your work.
It's an important question, because since the 1940s, we know that very small particles, and they found this with polio virus. They could actually see polio virus migrating up the olfactory. With particles, if you expose animals to particles, nanoparticles of manganese, you can actually see them accumulate in the olfactory bulb and then slowly diffuse into the brain. So clearly this is a route of entry that could occur. Also, nanoparticles deposit throughout the respiratory tract. And so they would be the most prone to affect the brain from that route. And to some extent, it's our data where we see a bigger effect on the rostral, which includes the near the olfactory bulb. That section is the first one to be hit and seems to have, the place where we saw the biggest effect. So it's certainly possible. As I said, when we do the size distribution on our particles, even though we call it PM2.5, a lot of them are submicron, and a very large proportion are down in the classical nanoparticle size. So they could be very important. However, it could also be material depositing in the lung, creating a large output of various inflammatory cytokines. And we know that, for example, interleukin-1 can migrate across the blood-brain barrier after it's generated in various parts of your body, usually because of an infection. And when the high levels of interleukin-1 migrate across the blood-brain barrier, it causes your body temperature to go up. It changes your temperature control. So the cytokines could be playing a role. And most likely, my guess is both things are happening at the same time.
All right. If there are no more questions, I'd like to thank Mike Kleiman once again for a wonderful talk. Thank you.
Thank you.
Thanks, David.